Alright, this last video over inheritance is going to talk about different types of dominance. The most common type of dominance that we talk about is simple dominance. Simple dominance is going to include your monohybrid crosses and your dihybrid crosses. So monohybrid crosses and dihybrid crosses are in another video which you need to watch. Now I want to talk about incomplete dominance. The first type of dominance that I discuss is not simple dominance, it's called incomplete dominance. When you say something is incomplete dominance, that means that the dominant allele is not dominant to the recessive allele. So there's a different definition. So we say that the heterozygote I'm giving an example. Is an intermediate in phenotype, which is physical appearance, is an intermediate between two homozygotes. Now, if you say simple dominance, simple dominance doesn't make sense for a lot of human characteristics. If that was the case, then we would have only short people and tall people. So in simple dominance for peas, you can say that homozygous dominant is tall, and you can say that homozygous recessive is short. Now, heterozygote of something that's simple dominance would be tall. But we're basically saying that anything that's an incomplete dominance is an intermediate. It's a mix. It's an intermediate phenotype. So what I mean by that is if you're a heterozygote, instead of being tall, you would be medium height. It is a mix. So there's no simple dominance here. The heterozygote is an intermediate. When you say tall and short, the intermediate of the heterozygous is medium. Now, if we said that we were doing this with animals, and you said that big B is black and little b is white, the mediate, if it was incomplete dominance, would be tan or brown when you mix black and white. So a very common example of this is going to happen in plants. So here's an example. Is if you cross we're going to say a red plant with a white plant, and it is incomplete dominance. The intermediate has to be in between. So when you mix red and white, the intermediate is going to be pink. So you cross the red plant with a white plant, and your offspring all end up being pink. This would literally be the genotype is a heterozygote, the phenotype is pink. The phenotype is an intermediate. When you cross a red plant with a white plant, you end up with a intermediate phenotype if it's a heterozygote. That's incomplete dominance. It's no longer simple dominance. Now, the next one, the next type of dominance is called CoDominance. when you say codominance, co means together. So neither one is dominant. Instead, they work together. Now, this is more common in animals. Now, an example, if you have a black dog who is homozygous dominant, and if you have a white dog who is homozygous recessive, your heterozygote will end up being a white dog with black spots. So it is both white and black. That's what codominance means. It is going to have both aspects. They're dominant to each other. So it doesn't mix like incomplete dominance. So if it was incomplete dominance, this would be a tan dog. If it were simple dominance, the heterozygote would be a black dog. So you have to look at the type of dominance. Codominance means that it is going to be both aspects. It has both phenotypes of both. So it is both white and black when you have the heterozygote. So incomplete dominance, they would be mixed. Codominance is both, and simple dominance, it would just be black. So we have three different types of dominance. You have to pay attention if it's simple dominance, incomplete dominance, and now codominance. Now, an important aspect of codominance is going to be the human ABO blood group. Now, when we talk about genes, your blood cells actually have a gene on the outside, which is called the I gene, which represents the antibodies. So to understand your genes, blood typing is on the I gene. So this would be a homozygous dominant blood because it's on two big I's. When you have a big I and a little I, that is heterozygous. And when you have two little I's, that means that basically you're lacking this gene, so you're homozygous recessive. Now, where the blood typing comes in, A and B are proteins. So these proteins are going to attach to your I genes of blood. So your blood type is on the I gene. So you can either be homozygous dominant, heterozygous, or homozygous recessive. So basically, you can either be A blood. So A is a protein that is attached to the I gene. So you can be A blood by being homozygous dominant A blood. That is A blood. But you can also be heterozygous A blood. Heterozygous is a big I, little I. So this is a homozygous dominant A blood. Now, the phenotype is A blood. This one is heterozygous A blood. So you can do the same thing with B blood. You can be homozygous dominant B blood, and you can also be heterozygous B blood. Homozygous, now B blood. Homozygous dominant because you have two big I's, or heterozygous B blood. Now, human blood type is an example of codominance. So I personally am AB blood. So I have an A on one eye and a B on the other. So technically, because there's two big I's, this would be homozygous dominant. Because there's two large I's, A, B blood. So it is both A and B, and that's what makes it codominant. But if you're O blood is homozygous recessive. O blood is the phenotype for homozygous recessive. Now, these are funny when doing a cross. So if I wanted to take myself, I am a homozygous dominant AB blood, and cross it with someone who has I blood, you do a normal Punnett square. But you have to separate your I's. So in this one will represent as me. I take my dominant allele that has the A protein and my dominant allele that has the B protein, and I separate them. For the O blood, I separate the little I's. Now, just like we did in regular crosses, you always keep your dominant letter in front. So, the first child would get a large I from me and a little I from whoever I have a baby with. Or they can get a large I and a B and a little I, an A and a little I, and a big B and a little I. So if asked what are my possible genotypes of my kids, they are going to be, two of them would be heterozygous. The phenotype would be A blood. However, two of them could get the B protein attached to the I, and two would be heterozygous B blood. So crossing blood can be a little bit different, but as long as you understand that our blood is going to go on the I gene, and there's different proteins, A or B, that it can attach to these I's, then you can understand the human blood group typing easily. But blood typing, because you can have both A and B blood, is an example of codominance. So those are extensions of Mendel. There's more in the textbook, but those are the ones that I would basically like to talk about. Now, there is one other type of inheritance that I really need to cover, but I will do it in a separate one.